Assalamu alaikum, guys, and good evening. Today, we are going to start our next topic in continuation of what we have studied in the central dogma of molecular biology, where we studied about DNA replication, transcription, and translation in relation to both prokaryotes and eukaryotes. So, this is actually the continuation of that topic, and the topic for today's lecture is regulation of gene expression. As I mentioned you earlier, that in case of the 411, where you studied about different metabolic pathways and different metabolic reactions for proteins, carbohydrates, and lipids in the 412, you have seen that all these metabolic pathways and reactions, they must be controlled and regulated. And this is very essential because the reason is that if these pathways or these mechanisms are not regulated or controlled, that's going to be a wastage of energy, and that's going to be a wastage of the intermediates, the products, and the reactant. In a very similar way, when it comes to the gene expression, it includes both transcription and replication and translation, because we are processing more DNA molecule from one DNA molecule, which is replication, and we are interested in the synthesis of particular protein, for which we need to go through the process of transcription to get the messenger RNA, or in some cases ribosomal and transfer RNA, and finally to get the translation process done to get the proteins. So this process must also need to be regulated, because there should be a mechanism that will determine that which protein is required by the cell or by the body at a particular moment. And for this, as we can understand, that there should be a mechanism which is going to regulate this process, and this is what we are going to study today under the title of regulation of gene expression. This is a little bit of complicated topic. I'll try to simplify this as much possible as I can, but always you have the time to go through the presentation after the lecture and listen to the recording, and then you can come up with a question anytime. So let's just start. The overview that we're gonna discuss in this is that the pro and the eukaryotes, they alter the gene expression in response to their changing environment. What actually the changing environment is, it can be an internal environment and it can be an external environment. What it means by internal or external, for example, if you're talking about a cell, there are some changes going on within the cell, and those changes are going to affect or alter the gene expression. In a similar way, there could be some environmental changes outside the cell which are going to affect the gene expression. So that's what I'm saying, that this sort of environmental changes can be internal or external, and they directly or indirectly affect the process of gene expression. In case of the multicellular organisms, where we have got multiple cells, the gene expression regulates not only the process of gene expression itself, but also the development of the organism, and is also responsible for differences in cell types. Like if you talk about the multicellular organism, we all know that when the cell division takes place, both the mitosis and meiosis, there is a process of cell differentiation. Like when a human zygote develops from two cells, one is from the ovum and the other one is from the sperm, that cells not only is multiplying, but it's differentiating into a specific type of the cells, like the neuronal cells, the nephronal cells, the muscle cells, the cardiac cells, all these kind of differentiation. So this cell differentiation is also an important part of gene expression, which we will be discussing in this, but not in that detail, because it's not exactly our point of interest at this point. But just to keep in mind that multicellular eukaryotes, their gene expression is also regulating the development and the differentiation of the cell types. RNA molecules play a very important role as well in regulating gene expression in eukaryotes. And again, this process or this topic we're going to discuss very briefly, but we'll not go in the details of how that happens. But just to be kept in mind that RNA is having a role in the regulation of the gene expression when it comes to the eukaryotes. Okay, so the first topic we're going to start today is about prokaryotes. We start our gene regulation topic first with the prokaryote, and then we'll go towards the eukaryotes. So what we're talking about right now is that bacteria, they often respond to different environmental changes, as I said earlier on. And these changes could be internal or external. And these changes can be a source of regulation. And what we are talking about right now is the source of regulation for transcription. What is transcription? The formation of RNA, messenger RNA specifically from the DNA. Now, what happens actually in bacteria, in contrast to the eukaryotes, there is a process of natural selection that has favored bacteria that produce only the product needed by that cell. Like, when you talk about the genetic material of the bacteria, a plasmid, that plasmid may have different genes, and these genes will code for different proteins. Now, the bacteria is doing a natural selection. That means it's going to produce only those products, only those proteins which are needed by the cell. In other words, not all the genes which are present on the bacterial DNA are going to be transcribed. This process is called the process of natural selection, because the bacteria is going to determine, with reference to its environmental impact, that how and when the genes of a specific interest must be transcribed and then later on translated. A cell can regulate the production of enzymes by feedback inhibition or by gene regulation. You have studied about the enzymes. Enzymes are protein in nature, we all know. And enzymes can be regulated at two levels. Now, the one level of the regulation of the enzyme is that you have studied in 411, like product inhibition, where a particular reaction catalyzed by an enzyme will be inhibited if we have excess of the product, because product itself will get a feedback inhibition to that enzyme that this reaction is no more needed. Similarly, we might have certain inhibitors or other molecules that are also going to affect the enzyme activity, like a competitive or non-competitive inhibition that you might have studied. But there's another level of the regulation of the enzyme, and that is at the level of transcription and translation. Like, because enzymes are proteins, so there is one level of regulation at the genetic level and one level of regulation of the enzyme at the cellular level. And both are equally important and both have correlation with each other. The gene expression in bacteria is controlled by a model called operand model. What is operand model? We'll go and discuss in detail. Now, this is the diagram in front of you, where you can see there is a metabolic pathway, a small pathway, I would say. It starts from a precursor. This precursor is converted to, I would say, product 1, catalyzed by enzyme 1. This product is going to be converted into another, which is called product 2, catalyzed by enzyme 2. And then this is going to be converted to its final product, which is product 3, or I call it tryptophan. So after 3 steps, this precursor is being converted to its final product, that's tryptophan. Now we have three enzymes involved in this pathway: enzyme 1, enzyme 2, and enzyme 3. Definitely, these enzymes will have been transcribed and translated from specific genes. So there is one level of regulation that I was talking about at the cellular level called the feedback inhibition or product inhibition. You can see more the amount of the tryptophan, it's going to affect or inhibit the activity of this first enzyme. Why? Because when you have got excess of the tryptophan available, you don't need that precursor to be converted into tryptophan. And that's what I am calling feedback inhibition or product inhibition. So this is at the cellular level. Now coming back to the other level that I was talking about, that is the genetic level. And at the genetic level, we can see that there are a couple of genes involved for the synthesis of these enzymes. For example, TRPE genes and TRPD gene is involved for enzyme 1. TRPC and TRPB is going to be involved for enzyme 2, or TRPB, sorry, not TRPC is only for enzyme 2, and TRPB and TRPA for enzyme 3. Now, if you have got excess of the tryptophan, which is the final product of this pathway, this tryptophan is also having a negative impact on the gene regulation or gene expression of these genes altogether, you can see. Now, this is also a regulation, but this regulation is at the level of genes, and that's called the genetic control. So this is the regulation of enzyme activity in short, and this is the regulation of enzyme production. And I'm sure that you can differentiate, because here we are not increasing or decreasing enzyme amount, rather we are affecting enzyme activity. While in this case, it's going to affect the enzyme quantity. So one level of control or the regulation is at the enzyme activity level, and the other one is at the enzyme production level, right? Okay. As I said that the prokaryotic regulation is run by an operand model. So what is an operand? What is the basic concept about the operand? The operand can be defined as a cluster of functionally related genes that can be under the coordinated control by a single on-off switch. So the definition is, I think, very much clear. We have a cluster of genes and they are functionally related. They're not different genes. They have some sort of functional similarity. And these cluster of genes, or this cluster of genes, is controlled on and off by one single control. So that means a switch on would result in the expression of all the genes in that operand, and a switch off would result in the termination or inhibition of the entire cluster. The regulatory switch which we talk about here is actually a segment of the DNA which is typically being called as operator, and this operator is usually positioned within the promoter region of the DNA. An operand is the entire stretch of the DNA, which is including that particular operator, which I mentioned right now, the promoter, where this operator is located, and the genes that they control. So the cluster of the genes plus the promoter plus the operator. This whole complex is called as operand. So I'm sure that you got an understanding what operand is. The operand can be switched off or switched on, as I said, by a single molecule. So the molecules that's going to switch off this operand are called as repressor, in contrast to this one, the operator. So this operator is basically the one which is promoting or activating the operand, while the opposite to that is a protein called repressor protein, which is going to turn off the operand model. The repressor prevents the gene expression or gene transcription, as I would say, by binding to the operator region. Once it binds to the operator region, it prevents the binding of the RNA polymerase. And once RNA polymerase is not there, transcription will not start. So you understand, this repressor is going to bind on the operator region, which is actually a location where the RNA polymerase should bind in order to start the transcription. And as long as the repressor is there, it's going to prevent the RNA polymerase to bind with the operator. And that's how it prevents the transcription. The repressor itself, as I said, is a protein. So it is a product of a separate regulatory gene. So, keeping in mind that repressor itself is a protein which is being transcribed by some other genes. So this is another thing to be kept in mind. The repressor can be in two forms. One is called the active and the other one is called the inactive form, depending upon the presence of certain other molecules, which I referred earlier as environmental changes. So the environmental changes are actually the presence or absence of certain molecules which are going to affect the repressor protein to keep it in its active or inactive form. And I think it's pretty clear that if it's in the inactive form, it's not going to bound with the promoter. And thereby transcription is going on. But when this repressor is in its active form, it's going to bound on the promoter. And thereby it prevents the RNA polymerase for its attachment. And thereby the transcription is going to stop. Now there is a concept called a co-repressor. A co-repressor is just like a cofactor. That means a molecule which is going to participate with the repressor in order to perform its function. So a co-repressor is a molecule that cooperates with a repressor protein, or it facilitate a repressor protein to switch on to switch an operand off. For example, if you talk about E. coli, E. coli can synthesize the amino acid tryptophan. The same example that I have discussed with you a couple of sites before, which was a generalized model, and now I will be going in a little bit of the detail. So I'm sure that you understand what is a co-repressor. The molecule that cooperates with the repressor protein in order to help that repressor to turn off an operand. Now, when we talk about the E. coli, which is the simplest model to understand, the simplest model to get the idea how the operand works. By default, the E. coli, they do have an operand called TRP operand, tryptophan operand, because as I said here, the E. coli can synthesize amino acid tryptophan. So that means they have an operand which is by default in there, and that's called the TRP operand. And that TRP operand in E. coli I'm talking about is by default on. What does that mean? It means that under the normal circumstances or a default condition, the genes involved in the synthesis of the enzymes which are required for tryptophan synthesis is in on mode. So generally those genes are transcribed, which results in the production of those enzymes and enzymes which involve in the tryptophan synthesis. If you remember the last slide I shared here, this one. So this is what I'm talking about in detail now. So when tryptophan is present, now consider the condition. When tryptophan is present, it binds to the TRP repressor protein. Tryptophan is the final product, remember. When you have tryptophan, it's going to bind with the TRP repressor protein. As a result of that, the operand is turned off. Why? Because here, the tryptophan is kind of a co-repressor. It's a product. It's a co-repressor. The tryptophan, if it's there, it's going to bind with the repressor, which is in turn going to bind with the promoter, which in turn stop the RNA polymerase to bind, and which in turn stop the whole process. So this is how a feedback inhibition is also being expressed. Now, the repressor is active only when you have the co-repressor tryptophan. Now, if you don't have tryptophan, the repressor protein is there, but it's inactive. But when you have more and more tryptophan within the medium where the E. coli is growing, it's showing that you have enough tryptophan. You don't need to synthesize tryptophan. So that means the repressor is going to be activated because its co-repressor is there. So the TRP operand is turned off or repressed if tryptophan levels are high. So you can see that with this example, we can explain both kind of regulations. The regulations at the level of enzyme activity, as I mentioned, because when you have tryptophan levels elevated, it's going to affect the enzyme activity, which is actually synthesizing tryptophan. On the other hand, this tryptophan is also working as a co-repressor here, and this co-repressor going to bind with the repressor, which in turn is going to stop the process of gene expression or transcription of these enzymes. So this is how the operand model operates, and this is one very simple example of TRP operand in case of the E. coli. I think it's so far so good. This is another picture of explaining the same phenomenon, but it's in the pictorial form. You can see here that this is the TRP genes, TRPE, D, C, B, A, which I mentioned earlier. This is the promoter region. This is the operator region. This is RNA polymerase. Now here you have understanding that this entire region is called TRP operand. Under the normal process, transcription is going on, and as a result of this, the mRNA is formed, and this mRNA formation will give rise to protein synthesis, and you got specific proteins E, D, C, B, A, which are specific enzymes, and that will facilitate the production of the tryptophan. In other case, when you have this inactive repressor here, you can see this inactive repressor would become active because of the binding of the co-repressor, and that co-repressor is in this case is tryptophan. So you can see here tryptophan absent, repressor is active. Tryptophan present, repressor is inactive. Tryptophan present, repressor becomes active. Once it is active, it binds to this region. When it binds to this region, it inhibits the binding of RNA polymerase, and thereby the entire process is stopped. This is the same diagram, just the second part of the diagram here is being shown in a separate slide. That's the same, nothing different. And we have some more diagrams coming in, which is just the repetition, so I'm not going to discuss them again. So this is how it's showing that the DNA give rise to mRNA, and this is specifically I'm talking about the repressor protein, because I said that repressor is also a protein which is being synthesized by a separate gene. So this is the product which is called the repressor, and once the tryptophan is there, it becomes active repressor. The active repressor is going to bind here, and as a result of this, the whole process is turned off. So this is how it is shown. Right. So far so good. We have discussed about operand model, and we have gone through one example of TRP operand in case of the E. coli. Now let's go in further detail to talk about repressible and inducible operand. What actually it means, repressible and inducible operand. There are two types of negative gene regulation. Why we call it a negative gene regulation? Because we are talking about the inhibition of the process. That's why we are calling it a negative gene expression, while the positive gene expression would be that means more transcription and then more translation. Now, a repressible operand is the one that is usually on, and a binding of a repressor to the operator shuts off the transcription. The example of a repressible operand is the one that we have just studied, TRP operand, because you have been told that the TRP operand under the default condition is turned on. So remember, a repressible operand will be the one which is under the default condition on, and it will be shut off if a repressor binds to this. Example, TRP operand. While an inducible operand is the one that is usually turned off, just opposite. A molecule in this case called an inducer going to inactivate repressor and in turn start transcription. So that's the process of induction. That means under this condition, inducible operand is the one which is turned off. Why it is turned off? Because it has already repressor attached to it, which is preventing the process. So what's going on in this? There is a molecule called inducer, and that inducer is going to inactivate the repressor. Once the repressor is inactivated, just you have seen in the previous pictures, it will be detached. Once it is detached, RNA polymerase can bind, and thereby the transcription can start. So this is an example of inducible operand. So I'm sure that you guys are now clear what is uh, an inducible operand and what is a repressible operand. You have studied about the TRP, which is an example of the first category. Now we're talking about the lac operand. Lac operand, which is a lactose operand, is an example of inducible operand. And now we see how inducible operand is going to work. This lac operand contains the genes that code for the enzymes used in the metabolism and hydrolysis of lactose. So that means these are the enzymes required by the bacteria to do the breakdown of the lactose. Lactose, we all know, is a disaccharide. So remember, this is an example of inducible operand. In the case of that, it is turned off by default. So that means transcription is not going on. Enzymes are not being transcribed and translated, which can hydrolyze lactose. So by itself, the lac repressor is active in this case. And as I said, this repressor is already bound with the promoter region. And therefore, this operand is by default turned off. Now, a molecule in this case called inducer, as I mentioned earlier, is going to inactivate the repressor, thereby detaches the repressor from the promoter region, allowing the RNA polymerase to bind on the promoter region and initiate the process of transcription. That means the operand is turned on. Now, this is the example. You can see here that this is the regulatory gene, which is going to produce the mRNA, and that's going to produce the active repressor. This active repressor binds here, so no RNA polymerase can bind, so no transcription takes place. And this LEC-Z gene is the gene which is there to synthesize those enzymes. So this is the situation number A, which is in absence of lactose. Because when the lactose is not there, obviously bacteria doesn't require those enzymes to break down the lactose. It makes sense. And that's why the operand is off. Now, what happens if you grow the E. coli in a medium, let's say, where you have lactose is the only source for them to multiply? Now, what will happen in this case? When you have E. coli growing in a medium where you have only lactose, that means the bacteria requires the enzymes to utilize lactose. That means you need to transcribe these genes. So in presence of the lactose, what will happen? As a result of that, it's actually not lactose, it's a similar molecule like lactose called allolactose. This allolactose will bind with the repressor, converting the active form of the repressor into inactive form. And as a result of that inactive form, it's not going to bind here, and thereby allowing RNA polymerase to carry out its function. And the transcription is going on. And as a result of the transcription, you have beta galactosidase, permease, and transacetylase, all the three enzymes synthesized which are required for the hydrolysis of the lactose. So again, it's the same diagram just being shown with a bigger picture, nothing new. This is also the same. So now you got the idea about repressible and inducible operant. Normally, inducible enzymes function in catabolic pathway, as you have seen in the case of the lactose. Their synthesis is induced by a chemical signal. And what was the chemical signal? Allolactose, or the presence of lactose, I would say. While the repressible enzymes usually function in an anabolic pathway, you have seen in the tryptophan synthesis. And their synthesis is repressed by a high level of the end product. So now I think it's pretty much clear that inducible enzymes usually function in catabolic pathway like lactose enzymes, and repressible enzymes usually function in anabolic pathway, just the example of tryptophan. So the regulation of the TRP and lac operant involves negative control of the genes because operants are switched off by the active form of the repressor. That is why we called it a negative gene regulation. So both of these regulations are kind of negative gene regulations. Now, coming back to the another part, which is a positive gene regulation. Now, this time we are talking about the other story, how the gene regulation is positively regulated. Again, the model is the same, the operant model, but this time instead of inhibitors or repressors, we're talking about activators. Now, some operands also subject to positive control. And this positive control is through some proteins. We call them stimulatory proteins, such as catabolite activator protein called CAP. This CAP protein is an activator of transcription. When glucose, which is a preferred food source of E. coli, is not there in the medium, that means the E. coli must have to look for the alternative source. For this, CAP is activated. And this CAP is activated by binding with the molecule of cyclic AMP. Now, this activated CAP is going to attach with the promoter of the lac operant. And as a result of that, what will happen? Increases the RNA polymerase binding, thereby accelerating the transcription to give production of those enzymes which are needed by the bacteria to hydrolyze lactose. Now, listen very carefully. If you look at this positive gene regulation, you can see that the outcome is same. Like in case of the inducible operant, what was the outcome? The outcome was the synthesis of those enzymes which are needed for lactose metabolism. And the outcome in this case is also the same. But the effect is different. In the first case, it was allolactose which was acting as a molecule to start the gene expression. But here, this is called the positive gene regulation, where a molecule called CAP is activated. Why it is activated? Because E. coli preferred source is glucose, and you don't have glucose in the medium. As I said earlier, if you grow E. coli in a medium where you don't provide glucose, rather than that, you only provide lactose. Now, what will happen? The bacteria uh, has no other choice to use lactose as a source of energy. And you have studied that lactose operand is generally switched off. So in this case, we need to activate the lactose operand. In this case, CAP is going to act as a promoter. It's acting as a kind of activator to bind on the promoter region of the leg operand and increases the affinity of the RNA polymerase. Now, these are two different things. In the first case, it was allolactose, which inactivates the repressor, thereby allowing RNA polymerase to bind. In this case, it's not like this. It is about increasing the affinity of the RNA polymerase to bind and thus accelerating the transcription. That's why it's called positive gene regulation. When glucose levels increase, automatically CAP detaches from the leg operand and the transcription returns to the normal rate. The CAP helps to regulate other operands that encode enzymes used in the catabolic pathway as well. So this is the example, again, diagrammatic illustration. You can say that this is leg I, promoter region, RNA polymerase is bound. That means transcription is going on. Now this CAP, which was in inactive form, becomes active. Active CAP will bind here, and it promotes the binding of RNA polymerase, or it enhances the activity of the RNA polymerase. So in this case, lactose present, glucose is scarce, CMP level high, more mRNA synthesized. While in this case, lactose present, glucose present, but CMP levels very low, that means little. So if you have availability of the glucose, no matter lactose is there, but you don't need much of the enzymes to utilize lactose because the preferred source for the E. coli is glucose. So that's how this regulation takes place, and the same picture again and again, just to give you an idea about the bigger picture. Okay, so we have completed the first part of the discussion, which is regulation of gene expression in prokaryotes. I'm going to stop here, and I want you guys to go through the slides again and listen to the recording again if you want to, and go through in details and try to understand how this gene regulation takes place in case of the prokaryotes. So we are going to stop here, and the forum is open for the question. If you have any question, just raise your hand. Yes, Nimra, go ahead.
Assalamu alaikum, sir.
Walaikum
As we
assalam.
discussed now that high the production of end product, so the operand will switch off. So my question
Yes.
is, instead of the end product, any other type of molecule will help in the initiation of stop, or in the or it will be help in the switch offing the operand?
No, first of all, you have to consider the first point. Uh, Remember the repressible and inducible operand. To keep it in mind that this operand is by default on or off. This is first thing. Your question refers to that operand which is by default on. That is the TRP operand. Now, in this case, yes, product is going to feedback inhibit, because that makes sense, because when you have enough product, you don't want much of the product to be produced. Regarding other molecules, yes, there is a possibility that some of the intermediates of this pathway, like for example, if the final product is produced after 5, 6, 7 steps, that means some of the intermediates of that pathway can also act like an inhibitor, just like the product itself. I hope that answers.
Yes, sir. Thank you.
Welcome. Any more questions, guys? Okay, now we are going to finish, and inshallah, I'll share the recording with you guys, and the presentation as well shortly. And see you guys on Monday for your practical. Thank you very much, and Khuda Hafiz.